Right. Hello. Is it good now? I hope so. Yes.
Okay. Can anyone let us know?
So I'll go a couple of steps backwards. So I was just looking at explaining. So platelets are in their resting state under physiological condition, do not interact with the endothelium. However, after vascular injury, you have the exposure of collagen. VWF binds to collagen by its A3 domain, and the shear forces of the blood unravel VWF, exposing its A1 domain, to which GP1B alpha on the platelet bind. Next, you also have a direct interaction of the platelets with collagen via alpha-2 beta-1 and GP6 receptors. GP6 binding to collagen trigger intracellular signaling events, leading to the activation of a very important integrin on the platelet, alpha-2 beta-3. And this is how platelets are able to bridge to one another via this integrin and the binding of fibrinogen that bridge platelets together. Platelets become more activated and release granule content. So you have release of VWF, release of ADP from dense granule that further activate the platelet and facilitate the recruitment of additional platelets. And the first layer of platelets, because of the direct contact with collagen, which is a strong agonist, change shape and spread to seal the wounds. Concomitantly to this, you have the coagulation that kicks in due to the exposure to the blood of tissue factor that lead to the formation of thrombin. Again, very strong agonist to platelets. And this is how further platelets get recruited, and you have the hemostatic blood that form, ultimately that is stabilized by fibrin, which is the cleavage product of fibrinogen by thrombin. So hemostasis is a very highly regulated process, and the importance of hemostasis is highlighted by the high incidence of cardiovascular diseases. All these diseases are fundamentally very different. However, they have a common denominator, which is the formation of a thrombus. And platelets play an important role in all of these diseases. There are many at-risk individuals that benefit from anticoagulant or antiplatelet agent. However, bleeding is a major limiting factor. So for the remainder of my talk, I will focus on venous thromboembolism. So what is venous thromboembolism? It encompasses two different diseases, deep vein thrombosis, and which is the formation of a thrombus in the valve pocket of deep vein, usually the leg. And sometimes this clot can break off and travel to the lung, which causes pulmonary embolism. And if untreated, this is a fairly high mortality, so 30% of people can die from pulmonary embolism. So one important point is that there is, as opposed to what I've just presented, there is no vessel damage, so there is no exposure of collagen in DVT. The procoagulant stimulus is intravascular, and it's a relatively slow process, so it takes days and weeks, and it is a process that occurs under low shear. This is to counteract like arterial thrombosis, which occur very quickly, the thrombus form very quickly and at high shear. So there's a number of risk factors associated with DVT, surgery, cancer, infection, long-term immobilization, such as long-haul flights. There's also genetic modification that lead to increased procoagulation, such as factor V Leiden. And usually DVT is treated by targeting the coagulation with heparin or more recently direct oral anticoagulants. And lately, there's very promising clinical trial targeting the contact pathway activation using antisense technology targeting factor 11. How venous thrombosis is initiated. So there is a very important component, which is the abnormal flow at the site of a defective valve. And the valve pocket especially constitutes a nidus for thrombus formation. It activates the endothelium, which also sometimes is stasis, so no flow, which creates a hypoxic environment, which also fuels this endothelial dysfunction. There is a VWF-dependent platelet recruitment to the activated endothelium that is needed to trigger DVT. However, there's no, as I mentioned, no denudation of the endothelium, so by itself, this is not the cause of thrombus formation. And this is where immunothrombosis come into play. So immunothrombosis is a relatively recent term that has been probably like in 2012, I think, the first record of this. It's to describe a physiological process where you have the formation of an intravascular thrombus in the microvasculature to limit pathogen spread. So it involves innate cells such as monocytes and neutrophils, and together with platelet activation, the coagulation and also complement participate to this. However, when you have on top of this inflammation, such as what is the case in DVT, that leads to aberrant immunothrombosis and the formation of a thrombus. There's also other cardiovascular diseases when this is happening, such as stroke or atherothrombosis, and more recently in COVID-19. So in DVT, leukocytes play a crucial role, and they get recruited to the VWF-bound platelet and the endothelium. Neutrophils are full of granules, and once they get activated, they release those granules. And for example, elastase can inactivate natural anticoagulant, TFPI, so that creates a procoagulant environment. And they also go under netosis, so the formation of neutrophil extracellular trap that are highly procoagulant. So NET provide a scaffold for red blood cells, platelets, and there's also VWF that have been associated with NET. And they also trigger contact pathway activation due to their negatively charged DNA and activate factor 12A. And lastly, if you remember, I mentioned like intravascular stimulus. So this is monocyte that release microparticles that are charged with tissue factor that trigger the extrinsic pathway of coagulation. And consistent with the importance of platelets and neutrophil role in DVT, the important role if you deplete neutrophil or platelets in murine models, or target VWF platelets capture, or target NET, all of this prevents venous thrombosis in mice. So how do VWF-bound platelets recruit leukocytes? There's a number of interactions that have been described. P-selectin PSGL1, CD40 ligand CD40, alpha-2b beta-3 binding to MAC1 via fibrinogen, or MAC1 directly binding to GP1B alpha and ICAM2. In all of these scenarios, one or both cells needs to be activated. However, if we think of the very initial events leading to DVT, maybe these receptors are not totally relevant because the platelets are not highly activated. So it's been known for some time that VWF-captured platelets can recruit leukocytes. And these are images from two different labs. So in this one, you have endothelial cells that are cultured in microvessels, stimulated to release VWF, and you can see those very long cable here in green. And here you can see platelets in purple on top of this VWF cable. And on top of the platelets, the blue dots are leukocytes. So from this, you can infer that platelet probably have a signal that trigger the neutrophil recruitment. And indeed, the GP1B alpha VWF interaction is capable of signaling. So when VWF bind to GP1B alpha, the shear forces of the blood pull the mechanosensory domain and trigger intracellular downstream signaling events, leading to the activation of SORC kinases, phospholipase gamma 2. There is release of calcium from the intracellular store, and finally, activation of alpha 2B beta 3. So the last few years, we studied this interaction, VWF GP1B alpha under flow, and interrogated if that could recruit leukocytes and also look at crosstalk between platelets and leukocytes. And this is my PhD student, Adela, who graduated now 3 years ago. So first, she expressed VWF-A1 domain and captured this onto microchannels here via the histag on copper pegylated surfaces. Blood is labeled with a lipophilic dye tau-6. And you can see the platelets in green are nicely captured on this VWF-A1 domain. And progressively, you can see the formation of microaggregates suggesting there's platelet activation. She looked into this, and indeed, you can see like the red signal here, which is fibrinogen labeled to fluoro4. And fibrinogen, if you remember from my slide, can bind to activated alpha 2B beta 3. And at the bottom, if you compare, when we perfuse blood on collagen-coated microchannels, collagen, which is a very strong agonist for platelets, the signal is much reduced. If we use an antibody against a platelet receptor that do not signal, PCAM1, you do not see activation of alpha 2B beta 3. We also look at the expression of P-selectin, which is also a marker of platelet activation. And we could not detect any P-selectin on VWF-captured platelets. However, on collagen-captured platelets, you had strong expression of P-selectin. So this is where the term prime platelet was born, to describe the expression on the platelet surface of activated alpha 2B beta 3, but with minimal P-selectin exposure. So the next step was to look at the recruitment of leukocytes on these prime platelets. And indeed, you could see here leukocytes that were rolling on this VWF prime platelet under low shear condition. I don't have time to show you the data, but we identified the platelet receptor, which was activated alpha 2B beta 3. So here you can see this is like a microchannel coated with activated alpha 2B beta 3, and you can see leukocyte nicely binding to the surface. We also we were interested in determining the population of leukocyte that was binding to these prime platelets. So T cells and neutrophils were able to bind to prime platelets. And you can see here, this is a very nice confocal, a video taken on confocal microscope, and you have like a neutrophil binding to a prime platelet and spreading and scanning on top of the platelets. There's also a heart shape formation at some point in that video. So we hypothesized that perhaps neutrophils were able to release neutrophil extracellular trap. So the next experiment that Adela did was to follow these neutrophils over 2 hours after binding to activated alpha 2B beta 3. So after about, I don't know if it's showing up very nicely, but after about 60 minutes, you can sort of see the nuclei becoming a bit fuzzy, suggesting DNA decondensation. And progressively, you have the green color appearing, which is a stain that stain extracellular DNA. So this suggested membrane permeability after about 85 minutes. To confirm that net were being formed, we used an antibody against cis-mutated histone 3, which is a marker of net formation. When we captured neutrophil with the anti-CD16 antibody, there was a significant reduction in net formation. Very interestingly, we also did an experiment where we passively captured neutrophil onto activated alpha-2b beta-3. And in here, you can see that there was, again, a significant reduction in the formation of net. So suggesting this mechanism is mechanosensitive dependent. So the next phase was to identify the receptor on the neutrophil. I don't have the time to show you all the data, but again, it was not MAC1 or not LFA1. And we collaborated with Dr. Grassi and Frontini from Cambridge, and that looked at RNA-seq database and selected candidate genes that were highly expressed in neutrophil and T cells, as opposed to monocyte and B cells. And they came up with a list of genes, and immediately one of the top targets was SLC44A2. Indeed, there was an article a few years back, GWAS studies identifying SLC44A2 as a risk factor for venous thromboembolism. They also describe a SNP of relatively high frequency that is responsible for arginine into glutamine substitution at amino acid 154. And individual homozygotes for this SNP had a 50% reduction in incidence of VTE. So to confirm that SLC44A2 could be our receptor on the neutrophil, we did two different approaches. One, we transfected HEC293 T cells with the wild-type SLC44A2 construct, fused with a GFP reporter, and we also transfected the cells with the R154Q mutant. And as you can see here, we showed that the cells were nicely binding to prime platelets. If we use a blocker to activated alpha-2b beta-3, there was a significant reduction in the binding. Or if we use a polyclonal antibody against SLC44A2, we could reduce the binding to prime platelets. Interestingly, when we use the R154Q substitution, then there was a significant reduction in the binding to prime platelets. The second approach we took was to genotype healthy individual. We identified a couple of individuals that were homozygotes for the protective allele. And when neutrophils were isolated from these individuals, similar to what we saw with the HEC293 T cells, there was a significant reduction in the number of interaction with prime platelets. So this could explain the protective effect that was seen in the GWAS study, and also perhaps highlight the SLC44A2 as perhaps a therapeutic target for DVT. So to summarize the first part of my talk, what I've shown you is that when VWF unravel on top of a cell surface, platelets can bind to VWF-A1 domain via GP1B alpha. This triggers intracellular calcium signaling, leading to the activation of alpha-2b beta-3. And you have recruitment of neutrophil via SLC44A2 on the flow. And this trigger net formation. So we hypothesized that actually this could be a very important interaction for the initial event leading to DVT. If you recall, so there's like we hypothesized that under disturbed flow condition, maybe VWF can get entangle and expose its A1 domain, priming the platelet. That would lead to recruitment of neutrophil and net formation and the formation of a thrombus. So how do we investigate the importance of VWF platelet priming in vivo? And so one approach was actually taken in vitro. So adding another component to the flow assays that I was showing earlier. So this is data from current PhD student Laura at Imperial College, who I co-supervise. So what she does is she culture endothelial cells and stimulate the endothelial cells so that they can release VWF and perfuse blood on top. And then you can see you have the platelet nicely decorating those cables of VWF. You also, under venous shear, you can also detect neutrophil getting recruited on these VWF captured platelets. When you use a blocker of the VWF-A1 GP1B alpha interaction, you abolished those interactions. And another cool thing she's been doing is to mimic the disturbed flow in those valves and using oscillatory flow back and forth. And then you can see that these VWF cable become entangled. You still see sometimes like neutrophil bound to these, but I don't have time to show the data, but she shows that these tangle are less prone to the cleavage by the regulator of VWF ADAMTS13. So they probably persist and create, again, probably could trigger thrombus formation. So these are like images. So the next thing was to show that indeed those neutrophils were able to form net under those conditions. And this was taken at the RAF. You can see in purple, so that's the DAPI stain of the endothelial cells. Neutrophils are in purple here, and you can see here net being formed on top of these cells. The next approach, we generated a mouse model with VWF GP1B alpha defective signaling, and we hypothesized that these platelets from these mice would be less prime, and therefore you would be seeing less recruitment of neutrophil, and hopefully these mice would be protected in a model of venous thrombosis. So to do this, we use CRISPR-Cas9 technology, introduce a codon stop to delete the last 24 amino acids of the GP1B alpha receptor. This is the summary of the characterization of these mice. They have a small decrease in platelet count. They have normal hemostasis, normal platelet aggregation. However, we did detect a defect in GP1B alpha and GP6 signaling. And this part of the research is carried on by Brianna, and she's looking at the crosstalk between those two receptors and platelets. So going back to our model here of microfluidic, consistent with the presence of the extracellular domain of GP1B alpha, you have a normal platelet capture in these mice compared to wild type. However, if you look at activation of alpha-2b beta-3, there is a significant reduction in the activation of these platelets. And consistent with this, these platelets have reduced ability to recruit neutrophil compared to wild-type platelets. We also looked at the dependency of this interaction, and similarly to the human scenario, if we use an alpha-2b beta-3 blocker, you reduce the amount of neutrophil bound to these VWF prime platelets on wild-type platelets. If you use an antibody against MAC1, we don't see any difference. If we use an antibody against SLC44A2, you reduce the amount of neutrophil bound to the platelets. And this is like new data with neutrophil isolated from SLC44 knockout mice. And there is also a reduction of the binding on prime platelets. So in summary, for this part of the talk, I showed you that GP1B alpha delta 6 platelets can be normally recruited on VWF. However, the signaling downstream of VWF GP1B alpha is reduced in the recruitment of neutrophil onto those prime platelets. The murine VWF prime platelet neutrophil interaction is alpha-2b beta-3 and SLC44 dependent and MAC1 independent. So moving forward, I hope to secure some funding to look further into the phenotype of the GP1B alpha delta sigmas in models of venous thrombosis. I will have PhD students starting in the fall, investigating specifically the mechanism of net formation after alpha-2b beta-3 binding. And I very much would like to also use these mice to look at perhaps the importance of this VWF prime platelet in model of infection and also other inflammatory diseases such as sepsis, stroke, and myocardial infarction. So to finish, I would like to thank the British Heart Foundation for the funding, the haemostasis lab at Imperial College London, especially Jim, Adela, and Laura, my collaborators, and few people at St. George's, Ian, Jody, Brianna, and the nurses, Vanessa, Maria, and the IRF Excellence Fund. Thank you very much.
Thank you, Isabelle. And you got in before I could ask a question about bacteria. Do we have any questions from the audience? Or online, actually? We can... There we go. Find them. Any questions? Yes, Julia.
So you showed that you have some neutrophils for patients with the SLC44A2 mutations. For these patients, did they have any sort of clinical signs of because the platelets and the neutrophils on top of each other? Surely there must be something.
So you meant in the GWAS study? No. So there is a new paper identifying actually homozygotes for the SLC44A2. So people that are totally deficient, for the SLC44A2, and these people are abnormal. They don't have any bleeding problems or anything. I don't know if that answers your question, but yes, I mean, this has not been looked at, and it probably would be very interesting to see, yes, what happens in, those individuals.
Chris?
When you said that the SLC44A2, is it a solid carrier for something?
Yeah, so it's a choline transporter, but it's a very bad choline transporter. So there's another isoform, SLC44A1, which is a lot better at it. Like I think it's 10 times better than SLC44A2.
So if you block that transporter to prevent this interaction happening, you don't necessarily affect some other aspect of metabolism?
I don't think so. There is a paper that highlighted the importance of SLC44A2 in platelets, in mitochondria. However, so choline mediated, but I'm not 100% convinced that the effect they saw on the platelet function could be attributed to that choline. Yeah.
Like a few mouse models you're working with, but I thought it would be nice to have a mouse model of that particular missense variant to knock in.
Yes, so that was my idea for a grant proposal, but unfortunately it did not fly with the reviewers. So yeah, I think this would be to me that makes sense, like to absolutely demonstrate that this substitution, that this amino acid is key to yes.
Great. Thank you, everyone. I think we're going to have to move on to the next talk, but thank you, Isabelle. I learned a lot. Sorry for those that were listening. I think we didn't have any sound to begin with, but thank you for joining us. And just a reminder that there are cakes at 2 o'clock, so come and join us in the collaborative space. So our next speaker is Kai Hilpert, and Kai has a very fancy presentation that I hope we're going to be able to share well. And so hopefully that's ready to go and working.
Sure.
Let's just move this one down into the corner. Is that going to help? So welcome, Kai.
Thank
Kai,
you.
You've been here for a few years. Like Isabelle with you. It's really good to hear what you're up to at the moment. So
Cool.
Thank you for coming to talk to us.
Yes. Well, thanks so much for inviting me to present, and I decided to give you actually 2 short stories. So one about peptides and the other one about floatation REST. So peptides I do normally in the lab. I work on it, and in my free time and on my own money, I investigated a bit more this floatation REST, and I thought it's on the point where it may actually get some benefits. Yes, so let's move on. Starting with the peptides. So, as you may all know, antibiotics are really important, not for only treating infections, but also for all kinds of operations that without antibiotics as, the precaution would not really work. And in the last decades, we have what's often now called the discovery void. So there's not much going on. However, what is going on is that the current antibiotics are starting to lose their efficiency. So in this paper here was shown that more than 1 million people die as a direct cause of resistant bacteria, and another 5 million where the death was associated to such bacteria. And that is obviously a number that unfortunately will increase even higher. So my work is centered around these peptides, and they're cationic and antimicrobial, and you find them basically everywhere in all living things, including humans. And we know about 3,000 of them, natural ones, and researcher created more than 15,000. In higher organisms, they're actually part of the innate immunity, between 12 and 18 amino acids long, cationic and amphiphilic. And they have different structures, as I will show you soon. Function-wise, as the name basically indicates, they do have antimicrobial or antibacterial activity. Especially interesting, they kill multi-drug resistant bacteria. And that's where we basically work on to target those that basically cannot be targeted anymore by normal antibiotics. They are also active against fungi. So we have done some tests against Candida and other fungi, and it works quite well. They can also be antiviral. There is a whole database of antiviral peptides, and part of them is actually coming directly from these antimicrobial peptide set. There's quite a lot of work done on malaria because they can actually also kill parasites. And more recently, it was shown that they can actually kill cancer cells. So with Ferrin, we have a project where we look at peptides that kill prostate cancer cells. And we just need to do one single experiment until we can publish that. As I said, in higher organisms, they are part of the innate immune system, and you can actually have a modulation of chemokines, cytokines, but they can also be chemotactic as well. They have different structures. You can see here it's a classical helical structures, but you also find beta sheets and obviously also combined structures. Next to their diversity in structures, they have quite a bit of targets. And that's really cool because you can develop many different peptides into the clinic and they will not interfere with each other. The other thing, if you really look closely, is that some of these peptides actually do have multiple targets at the same time, which makes it even more complicated for bacteria to develop a resistance. And that is obviously a big plus on these. So how do we go on analyzing these peptides? So Ronald Frank in Braunschweig has developed a technology, it's called spot synthesis, and you can see in this automate here does spots on a surface. We use cellulose, and with that we can basically synthesize 600 peptides in one go. So all these blue dots there is one single peptide, is each dot. And it gives you enough material to do various experiments. But if you do want to do, let's say, an antibody epitope scan, and you just need small amounts of peptides, we can actually synthesize 3,000 peptides in one go. So it's a really great technology, especially if it allows you to synthesize many peptides for a very reasonable price, which you otherwise wouldn't do. So once the synthesis is done, you need to punch them out, put them into micrometer plates, and then dissolve them, and off you go. You can then incubate them with your target strains that you are interested in, do a live dead stain, and then analyze the data. The same, because you have enough material, you can actually do a simple lysis assay, which gives you at least a hint of toxicity, and you throw everything out that kills basically everything. So what have we done so far in the last 20 years? So I started off actually with natural peptides that I optimized, and then I started de novo designs, which wasn't really so successful, and then switched to knowledge-based design, which was really good. And then because we have synthesized now more than 10, 15,000 peptides and screened them, we have so much data that you actually can do quite fancy bioinformatics with it, including artificial intelligence. And in our latest project, Nathan was doing that. We used genomes, whole genomes, and subtracted peptides basically also in silico, and then synthesized and tested them. We looked at their efficacy, how fast they kill, and we had various targets. Really important toxicity. So we did hemolytic, then we looked at different interaction with blood cells, because obviously you need to inject them. They are not overly available without any tricks, at least. We looked at human cells, waxworm, zebrafish. Stability, peptides, as proteins, they will be cleaved by proteases. So you need to put some effort into making them more stable, which actually was quite straightforward, I have to admit. That was fast. We put them also in a frame, in a scaffold that basically allows them, we can cook them, and then they fall back in their activity. So we thought it would be interesting for countries where, you don't want to have cooling all the time. So at least at 40, 50 degrees, you can store them and it doesn't matter. Synergy is quite an interesting point, because you can actually revive old antibiotics that don't work anymore. And if you add the right peptides, they can actually do work, which opens up a new avenue. Modifications is lipids and sugars. We tried that, it worked. Mode of action, obviously really interesting. Many people still believe that most of these peptides just make pores. The opposite is actually the case. The minority of them makes pores. Many of them go actually inside. We showed that they can bind to ATP, RNA, DNA as well, ribosomes, and we developed a whole new system to screen for their mode of actions. Because we have so many peptides, and we didn't know which one to move forward, so it was actually important to develop a method to screen for activity. I have given a talk at Nature webinars for Biosacs, so you can have a look if you're interested. It's a whole different story. So, and now I make a jump away from peptides to the floating. So I just want to give you a few impressions on depression, anxiety, and pain, and why that is maybe interesting as well in general, but also here at St. George's. So according to NHS, we have now 1 in 4 from the 17, 19-year-olds population have a probable mental disorder, and that is an increase from 1 in 6 just the year before. So it's a big jump. And what that means is that those people actually will be our first-year students in the next year. And then I looked in the national statistics from 2021 here in the UK, and you can see that the first-year students, and that's like overall faculties and things, have more than 35% of them have a moderate or severe depression or really high anxiety. So this is basically the challenge, I think, which needs to be somehow met. There was this wonderful study here where they looked at 198 different studies on this topic and found that the average of depression in medical students is 27%, which basically fits our model with these 30 that you saw before. And what I also was quite shocked about was only 15.7% of these students who screened positive for depression reportedly sought treatment. And then I looked a bit more on that, and I found a survey and where they had 68% of the students experienced mental health pressure during their medical degree, yet 50% say they would have concerns about seeking support. And 59% also think medical students and doctors are unable to seek support for mental illness without a risk of stigma. So not having really high amounts of depression and anxiety in that cohort, if you want. They're also not really wanting to look for help if they actually need it. And if you then look, this paper went to this suicidal ideation, which then obviously leads to suicide eventually. And the average they found is 11%, so that's quite high. This study here is actually from Nepal. You can see they actually did studies for each single year in this medical education. And you can see around, again, 25 to 30% struggle with depression, a little more anxiety. And then they looked at burnout at this Copenhagen burnout inventory scale. And you can see that by this year, when they go basically off to the clinic, around 60% have signs of burnout. So there's a big problem, in my opinion. And I just make a little jump again. And so it's pain. And as you may heard, there's this opioid crisis, especially in the States. And so there's about 10 million people in 2019 that misuse prescription. It's quite significantly high in the US. The UK is somewhere here in the middle, but there is a clear upward trend. So this is the rate of drug misuse death in 2021 in England and Wales, and you can see the numbers are just going up. So, floating, what is it? So the guy here on this picture is John C. Lilly, who wrote this book. He is actually the inventor of floating, but together with some of his colleagues. And for them, they actually wanted to explore consciousness. So they developed this situation where you lay in this chamber, let's say, and then the light goes out. You are in the dark. You don't hear anything. You don't smell anything. You don't taste anything. You don't touch anything. There's no gravity because you're floating, and there's temperatures exactly as your body temperature. So all the normal information that your brain needs to access is basically suddenly zero. That gives you a lot of capacity to go into yourself. And then something interesting happens. And So there was this review which I was quite amazed about. So the special forces actually looked in that and found out that this technology can actually be help improving pain, sleep, and performance measure in trained, untrained, and healthy adults. And they basically analyzed more than 35 studies that has been done on this technique. So, and I thought it's quite an interesting part that even the Army thinks that's something useful to have. In research, it looks a bit fancy like that. So you can actually measure all kinds of things, blood pressure, heart rate, even brain activity, and then really find out what's going on in these situations. And one, I think, normal thing for relaxation is that your blood pressure drops, and that's exactly what happens. It has a huge Cohen's d factor of 0.8. And this is just one float. So just laying in that tank for an hour, and you can see that all the things you don't want to have, like pain, fatigue, depression, stress, anxiety, goes down a lot and significantly a lot. And everything you do want to have, being refreshed, relaxed, peaceful, high energy, is actually going up. So there was one study which was quite interesting. They looked at generalized anxiety disorder. And it's quite tough to treat, actually. What they did, they had, I think it was 12 floats. So they went into the tank 12 times. And what they found is, you can see that here, the baseline was 10, and it went down to 7. Whereas in the control group, the baseline was 9.9 and went down to 9.2. And they did a study after 6 months, which showed that they actually went even better after that. So it went down to 6, which is quite amazing. And what I also want to point out is, whereas people in the control group who had psychotherapy actually stayed there, whereas in the treatment groups, some people at least left. So it shows that it can actually be really support the classical things you would do, psychotherapy and medication. And so my question was now, when I go to the floating centers, they're obviously all the same people, and I talk to them, and I got really interested why they actually go there and pay money and not a little amount to see that or to experience that. And so I did a survey, and I asked them basically, for how long are you floating? And you can see from the beginners to more than 10 years, everybody was in that group. And here's an interesting one. I asked them, how many times do you actually go floating? And the majority said 1 to 3 times a month, which makes sense because the best deals you can get is basically a membership with one float a month. And then I asked them, if time and money wouldn't matter, how many times would you go? And they said the majority then that 1 to 4 times a week, and some people even want to go daily. So there is actually something people really gain from it and find it fantastic. So the length of the float is also 60 minutes. It's not surprising because in these deals, the standard float time is 60 minutes. I was quite surprised that nearby 1/3 actually pays more to have the 90 minutes or even more. So what's their main benefits? Relaxation, stress reduction, obviously, sleep improvement, anxiety, burnout prevention, things like that came up with really high values. And the next really important question for me was, how long does the benefit actually last? And so many said 1 to 4 days, but some also said just a day. Obviously, this is really important if one thinks on developing clinical trials to know what's the best interval to use for that. I also asked him, what else did you do? You may not just go floating for your well-being, sorry. So most of them did walks in nature, meditation, breath exercises. That got me really excited because some of you may know, 20 odd years ago, I made a 3 years training to become a yoga teacher, which I then did for 20 years. And I also did Tai Chi and martial arts for, 15 years. And I got lots of training in what's called traditional counsel, and also got certified in a method which is called family constellation therapy, which was discovered by this guy here, Hellinger, which also looks at problems in anxiety depression area. So, but all these techniques basically have one element together. There is meditation and breath exercises. And I thought, well, maybe there's a good call to combine all that and make something even more effective. I also asked them side effects. Most of them didn't experience any. There was some dry skin and things like that. And then I asked them, what would you recommend if I do research? What should I do research about? They said basically stress, improvement of sleep, pain management, PTSD, burnout, anxiety, depression. So all the things that, have been shown already. They just, thought there is more to it. And with that, I want to thank all the people involved, the funders, and I thank you for your attention.
Thank you, Kai. So we've got 2 topics here. Do we have any questions for Kai on peptides or on floating? Chris?
Just about the peptide. So,
sure.
In terms of like therapeutic application, are there many that have gone very far?
Yes. So every single year comes a paper out with as a review about how many peptides of them are in clinical trials. It's always a lot. So far, there was only one that got approved for a really niche thing. So everybody in the field is still waiting, for that breakthrough. It's a little bit like the RNA technology with vaccination. They had like 25 years, nothing happened, and then the right moment. Yes, so they do produce quite a lot of clinical trials, and I'm really hopeful for 2 of them currently. So we will see.
When you are designing those,
sorry,
are you designing them with like specific protein targets in mind, or is it always just based on
me personally, or in general? Either. Me personally, not, because we do whole cell assays, so I don't mind whatever is killed as long as it gets killed. Some people, like the industry, has in the 2000s has spent millions into designing fancy molecules that bind to some important enzyme in the bacteria. And I think from, I don't know, it was 100 million they spent, and they found one because the binding and everything was great, but they couldn't get it into the bacteria. So, and I thought I don't want to do the same mistake again. So I stick to whole cell assays. Yeah.
Debbie?
Can I just ask about the floating?
Yes, please.
Sounds lovely, the thought of floating in some water, it's nice and warm, and taking your time out from your day. And is that where all of these things are related? It's actually taking time out, quiet time out of your day to contemplate, to, taking the stress out of your day. Is that not what the key thing is that's happening here that's making people feel the benefit?
It is, yes, a part of it. My personal experience is that I try to teach people meditation because it has shown, in multiple thousands of studies the benefits. But if you have somebody who has anxiety, depression, or whatever, and you say, sit there for 20 minutes and clear your mind, they just go bananas. Whereas if you lay in a tank, what happens is, because everything is floating, suddenly, for maybe people told me for their very first time in 10 years, they actually felt relaxed again, because all your muscles after 45 minutes, an hour, start to relax, because you don't need to hold anything, you float. And often what happens is when your muscles and your body starts to relax, your mind follows. So you have all these circling thoughts about, oh, should eat this and should that, and how bad is this, and when your body starts to relax, it is actually that sometimes your brain starts, not always, but often your brain starts to relax, you become in a natural meditative state, and it's so much more relaxing than I don't know what I compare it to watching TV or listen to your favorite music. It has not the same effect, really. It is part of it, as you say, for sure. Taking out an hour is great, but taking out an hour in a way that all your muscles relax, that your blood pressure drops, that your heart rate drops, your breathing frequency. In the brain, there's lots of things going on, and that combined gives you this kind of feel of wow afterwards.
It was related to the first part of the talk, the peptides. So in your screening, you use peptides to see their effect on directly killing bacteria. I was just wondering if you have, what happens if you use cells in combination to see if it has a synergic effect, or that, for example, macrophages or other cells that would benefit from the peptides. And
yeah.
Because I recognize one of the peptides, L37,
yes.
Is an activator of platelets, for example. So it primes the platelets to recruit human neutrophils, etc. So I was just wondering.
Yeah, I know that people have tried, but I didn't. I was more in the impression I still need to do some work on the fundamentals to get them towards clinical trials. This is where I just want to go. But we did test quite a lot of things with blood platelet activation. We did test with I think 10 of them, also the whole activation of pathways for coagulations and things. So you have to be careful there, as you say. Some of them do stuff that you didn't expect them to do. So yes, that's really cool. It's really good to test them.
I have a question, probably a silly question about flotation.
Yeah.
So it's flotation in water,
yeah.
right?
Yes.
What happened if you would suggest, I don't know, like people that go into space, no gravity, like you're floating as well? Do you think it would be the same effect than the flotation in water?
I never was in space, but I can imagine. I mean, if you are in the space station and you float, you close your eyes, you have some earplugs, and it's more or less the same thing, right?
Yeah.
So, but it's not so accessible.
More expensive.
Yes.
So maybe they don't have any problems with depression and anxiety.
Okay. Thank you, everybody. Thanks for coming. Thanks for your questions. And I hope to see you in the plastic space for cake in a few minutes. And thanks to both of our speakers, to Kai and to Isabelle.
Thank you.
See you next month.